I'm conscious that it's now time for coffee, and I think we have to not worry too much about the time while we get the talking done. I'll be as quick as I can, then we have a break, we'll take a photograph of everybody to prove you were here for your funding agencies, I know, just to have you all on record, and then we will go and have a tea break, and then we'll come back and hear from the clinical side more than the physics side. But so what I want to do is kind of explain why we're here talking about this at the launch, as opposed to, say, talking about some other physics aspect on health. And really, the whole thing came about as a result of a collaboration that was building organically. And this collaboration that was building organically was really between Brendan and Luis and others in the kind of medical physics radiotherapy zone directly. And it struck me, as I was head of school at the time, as a model of how to proceed into the future. So what I kind of see before I go through the slides is that's a kind of an example of how the process works. And other disciplines inside physics, and particularly the biophysics side and the imaging side, can feed into, and the space science, even the detector development side, can feed into other projects which slowly but surely move to the clinic, or maybe actually not that slow. So this is the school as I sort of see it. There's about 28 or 30 people associated with the school, and I say that because one or two of them haven't got here yet, and there's 0.2s and 0.6s and all this sort of stuff, but there's about 30 faculty associated with the school, and the ones in red are people who I've kind of associated with the Centre for Physics and Health and Medicine, and the ones in black are people who I haven't, but that doesn't mean they won't be or can't be, or haven't, it just is as it is right now. And the key techniques we use in the school would be imaging, simulation, and spectroscopies, and behind all of that sits kind of the increased use of data. And the kind of, if you look at the traditional areas in which we do research, we have atomic, molecular, and plasma down here. I forgot to put in plasma. We've got nanoscience, we've theoretical physics, we've particle and radiation physics, we've space physics, astrophysics. So these are the kind of key strengths that we would talk to the physics community about, but really the techniques are broader than that. And this is the sort of the sample pathway then. We have some sort of a collaboration which organically started, and it's based on maybe 20 years or 30 years of co-teaching in master's projects and PhD co-supervision and stuff. But it came more to a head in the last decade, and it's developed now to where we've got sort of joint positions. People are, like Brendan is 20% School of Physics and 80% St. Luke's. We've kind of we bought out his kind of left arm and his right knee and his head, basically, And then if we look at Sean, we've bought out more of Sean, and he's the director of the centre. And so these posts then, we've jointly supervised PhD posts. We've people who are permanent medical physicists doing PhDs part-time. All that sort of stuff has grown. And then the next bit is the centre, which we've got, and then move to kind of joint grant applications and a longer-term strategy. And I think Uwe's talk really teed up that we need to be kind of creative and imaginative in that process, not just incremental. I love incremental science. That's what I love doing. But I know that you got to take a jump or a leap. So, for instance, we've got this idea. Ronan is after. He's got a suitcase with him. It's a little one. He's very proud of it. He got it yesterday. It's a Medipix detector that was developed by a Synergy team out of CERN. And it does all sorts of interesting things. But this is really what it does. It's got a pixelated, energy-resolving detectability out to 140 kV. And it sits in it looks like a USB stick to all accounts, and it plugs into a laptop via USB cable. It's about 10 grand. It's really very nice. And he's going to show you that at coffee with the little radioactive source he'll put on top and show you that it works. But the potential for that particular device is demonstrated by the fact that himself and Brendan and others put it in the LINAC in St. Luke's, and they are developing a paper on its responses over time. I mean, not this particular one because that arrived yesterday, but a previous one that they got a loan of. So this sort of stuff is kind of the way we can see collaboration between particle physicists and clinical tools and clinicians into the future. And all sorts of projects spring to mind there. The plastic phantom would be could be very well studied with this, and then maybe the human also. So then this is a future pathway I think about. We've got an awful lot of biophysics research. You've heard from some of the biophysicists. I'm just going to skim through the others, and the slides are really there to look at afterwards. That could lead towards industrial drug food applications, joint grant applications, and out of here could come clinical activity. Because the biophysics, typically biophysics is a basic research, nanobio or biophysics is a basic research with orientation and direction and applications downstream. And it's very hard to see instantly how it could connect with clinic, but a joint application might be successful in funding terms and in research development terms. So then we look at the different groups. Brian Rodriguez has a group in nanoscale function, which really is liquid AFM, following on from work. Susie Jarvis brought the group, Brian joined it, he leads it now. And They do all sorts of interesting things where they can image in a liquid environment and study the liquid-solid interface in particular. They can look at cells adhering to surfaces. They can look at the elasticity of cells, the strength of cells. They can change you can genetically alter the cells or treat the cells and then go back and do it again. You can see all the different things. One of the things that stands out here is this measurement of the tearing forces in an amyloid fibril. You can look at all sorts of different properties, elasticity, surface charge, chemistry, all sorts of things, how they influence the behavior of nanoscale objects on a surface in a liquid environment, and presumably in a dry one too. James Rice, he does nanophotonics and nanoimaging, different kind of analytical techniques. Again, we see there's spectroscopies, typically microscopy and spectroscopies, sensitive to different types of materials, different nanostructured elements like, again, amyloids, for example, or sensitized nanoparticles. Possibly the idea to deliver light-activated therapies into specific regions of the body, which could then be irradiated with heat or light to activate them. We've got Donald McKernan, who's a researcher associated with the center, with CCAM, which is a molecular center, and an ECAM grant, molecular simulation. In this case, Donald's work has started making this sensor, which is based on a hinge which oscillates open and closed in the presence or absence of a particular thing you might try to diagnose. I can't remember the exact word now, but say the presence or absence of a chemical or a biochemical of a particular type of molecule. Could protein, for example, could cause this to switch on and off, and that, because it would have a flashing nature, only active in the presence of this particular biological agent, would be very sensitive to small quantities and would have a high signal to noise. And so there's patents filed here. There's been an experimental proof of concept, and it's on its way to hopefully an assay and diagnostic tool development. If you look at Vladimir, he does nanofluidics. Vladimir has a large group, large grants, lots of activity, but the bits that stand out to my naive non-nanoscientist mind, things like there's a very strong modeling component of the bio-nano interface, how cell membranes work, et cetera, et cetera. Toxicity of nanomaterials. These materials can penetrate the cell membrane and alter the behavior of the cell, and it can be toxic. And it just struck me nicely that could be imaged, for instance, with the microscopies that you've heard about earlier on. Things about biocompatibilities of implants, because implants can degrade into nanoparticles, which then can penetrate the cell membrane and cause all sorts of damage. Metal on metal is a particularly bad one. Then we look at things like how you actually manipulate the cell and maybe make it so that you can enhance the properties of the cell using nanomedicine. So how do you get nanocarriers to penetrate the body better? So this is sitting based on a computational simulation analytical approach in theoretical physics, moving into different targets experimentally. If we look at Antonio Benedetto and Pietro Belloni, Antonio Benedetto is he studies cell membranes using a combination of simulation and neutron irradiation, neutron scattering techniques. And so he is working, developing a new neutron spectrometer, but also looking at the room temperature ionic liquids, which are kind of complicated salts, which are liquids at room temperature, which have new properties. There's a big range of these salts. They can be magnetic, they can be large or small, they can change the behavior of membranes and the rigidity of cells, which he's investigating, going back to Brian Rodriguez's slide, taking these cells in the presence of room temperature ionic liquids, putting them down, measuring their mechanical properties, for example, the elastomechanical properties, then putting the magnetic liquids in and watching how a magnetic field may change the behavior of the cell on a surface. And Pietro then is the computational, intensely computational side of this, looking at different biological systems, how bilayers break down, again, ionic liquids, the fibrillation kinetics, looking at how drug complexes interact with tissue, possibly also protein folding, amyloids, those sort of things. You see them here. So again, amyloids come up from different approaches, spectroscopies and simulation. VO, a biophysicist simulator, again, theoretical computation biophysics, looking at all these different sorts of systems, Alzheimer's, amyloid fibrils again, protein aggregation and folding, how things attach, how cells adhere, how molecules interact with cell surfaces, how the lipid bilayer is interacting with different proteins in a kind of high-performance computing approach. Connected a lot to work with the National Institute of Health in the US. So literally I flew through it, and hopefully I didn't go too fast on one and too slow on another. The thing I suppose we want to think about for the afternoon, and you talk about this at coffee, is the strategy for the centre. You know, there's a thought MSc coming. We want to grow the PhD, the residency programs. We want to hire people out into the centre. We want to hire ad astra fellows perhaps, which I'll explain. Maybe we can talk about during the roundtable session afterwards. Maybe look at cost actions, look at individual research and academic funding, go beyond horizons the next EU round. And then the pipelines are kind of interesting. The universities at the hospital and the physicists inside the university to kind of H&M, it's not the retail outlet, it's health and medicine applications. And then potential funding channels, we'll talk about those later, but I just put them up so you'd have them on the slide when we go looking at them later. PRT Life 6 may or may not happen. It worked a lot in PRT Life 5 and 4 were successful, useful in UCD. And finally, the other thing to think about is this. We're standing in a refurbished phase 2 of science in the lecture hub, and where you had coffee was the east, was a new building. But west and north are still in their 1960s state, and they'll be refurbished over the next 4 years. And it'll add about 5,000 square metres of new space and about 15,000 square metres of refurbished space. And that'll be an opportunity for interdisciplinarity in the whole college. But particularly for us in physics and our collaborators in science, we'll be looking at the Centre of Physics, Health and Medicine as being part of an integrated part of this whole new phase 3, which will complete out science and give us about 67,000 square metres, which is massive space. And I mean, space, here's the third type of space. We've had it on the nanoscale, the astroscale, and this is the intermediate scale somewhere. It matters because we're going to try and grow this faculty in this college by 30% and the students by 20%. And that's opportunity. That's what that is. It's an opportunity to change what you do, hire new people, look at new disciplines. And this centre should inform some of those things, and it already is, along with other centres. So that's really it. Questions we better keep for coffee because I'll be shot. Thank you very much, and that's what we were supposed to be doing in the first place. Thank you.
So we'll